a diverse and engaged audience. Your participation greatly enriches our discussions and underscores our shared commitment to advancing in our respective fields. I look forward to productive conversations and collective learning as we embark on this educational journey together. SPSR is distinguished for its engaging webinars, further enhancing its status as a leading platform for continuous professional development in the pharmaceutical sector. I am confident that today's session will uphold our tradition of excellence, providing valuable insights from a valuable speaker. Let us seize this chance to gain new knowledge, participate actively, and progress our field. Welcome, everyone. I look forward to a productive experience for all participants. Thank you for joining us today. We are gathered today to commemorate the 96th SPSR webinar on Revolutionizing Eye Care Nanoparticulate System for Enhanced Ocular Drug Delivery, a distinguished event hosted by the Society of Pharmaceutical Sciences and Research. Under the esteemed guidance of Dr. Shashi Alok, National President of SPSR, the Society continues to play a pivotal role in fostering innovation and advancement in the field of pharmaceutical sciences. The SPSR is devoted to advancing the pharmaceutical field through a variety of strategies. Our mission is to organize conferences that support pharmacy-related initiatives and endorse programs tackling public health issues in India, focusing particularly on disease prevention. We make it a priority to recognize and honor pharmacists whose notable contributions drive progress in our field. At the heart of our mission is a commitment to spreading pharmaceutical knowledge, ensuring a steady and robust exchange of information about pharmaceutical practices and services. By striving towards these objectives, we aim to elevate the quality and impact of pharmacy profession, fostering a proactive and collaborative community dedicated to excellence. It is our great privilege to introduce Dr. Himanshu Pandey, Director CTE and Deputy Registrar, Central University of Higher Tibetan Studies, Varanasi, UP, India, as a distinguished speaker. Dr. Himanshu Pandey's expertise greatly contributes to our event, providing substantial knowledge and insights. His participation is certain to enrich our discussion and expand exploration of nanoparticulate system for enhanced ocular drug delivery. We are grateful for his engagement and anticipate an enlightening session. It is our privilege to introduce Mrs. Monika Sabharwal as the convener for today's webinar, who also serves as the founder and National Secretary of SPSR and CEO SPSR Theoreticals Private Limited. Her significant involvement and leadership in the pharmaceutical sciences and research communities are indicative of her commitment to the field. We are very grateful to have her guidance in today's session. Now we will start with the short introduction to guest profile. Dr. Himanshu Pandey is an academician and researcher currently serving as the Director in charge CTE and Deputy Registrar Administration at the Central Institute of Higher Tibetan Studies, Sarnath, Varanasi. With a rich academic background, he previously worked as the Associate Professor in Pharmaceutics at the Faculty of Health Sciences, Allahabad, where he made significant contributions to teaching and research in pharmaceutical sciences. Dr. Pandey has an impressive research portfolio with over 60 publications in reputed national and international journals and 8 published books and book chapters. His research expertise and dedication have earned him prestigious recognition, including the Young Investigator Award from the Department of Biotechnology, Government of India, and the ACS SCGS grant from the American Chemical Society, Washington, D.C., USA. He has successfully led 3 externally funded research projects with a cumulative value of rupees 1 crore and has supervised more than 11 PhD scholars in various areas of pharmaceutical sciences. In addition to his academic contributions, Dr. Pandey is actively involved in the policy and innovation initiatives. He serves as the mentor under the Atal Innovation Mission of NITI Ayog, New Delhi, and has been appointed as the assessor by the National Assessment and Accreditation Council, NAC, Bangalore. His administration acumen is reflected in his roles as a chairman and member of several important committees and selection boards at the institutional and national levels. His exceptional achievements have been recognized with numerous awards and honors, including the Chancellor Gold Medal presented by the President of India, Dr. APJ Abdul Kalam, as well as the Rajiv Gandhi Young Investigator Award, Young Scientist Award in 2014, and fellowships from renowned scientific bodies. Dr. Pandey also contributes as a reviewer for leading scientific publishers such as Elsevier, Springer, and the Royal Society of Chemistry, Cambridge. Dr. Himanshu Pandey continues to inspire the academic and research community through his multidisciplinary work, leadership, and commitment to excellence in education and pharmaceutical innovation. It is with great satisfaction and pride that we declare that we have exceeded the 1,300 registrations for this webinar. Registration received are from India, Bahrain, Ethiopia, Indonesia, Iraq, Kenya, Kuwait, Malaysia, Morocco, Nepal, Nigeria, Oman, Philippines, Romania, Saudi Arabia, Seychelles, Syria, Tanzania, Turkey, Vietnam, and Zimbabwe. We are deeply moved by the substantial and varied responses we have garnered. Our heartfelt gratitude is extended to each person who has taken a moment to participate in this significant event. Thank you all for joining us today. Let's make this webinar an insightful and enriching experience for everyone involved. We have our esteemed keynote speaker, Dr. Himanshu Pandey, with us. Welcome, sir. Your insights would be highly valuable, and I would appreciate if you could present them in a speech. Over to you, sir.
Thank you, Dr. Pradeep, for such a nice introduction. And I would, first of all, I would like to thank Dr. Shashi Alok and Dr. Monika ji for inviting me and giving me an opportunity to present in front of you all. And I am very happy to learn that not only from India, but number of participants has get registered throughout the globe. That is a good thing. And Society of Pharmaceutical Science Research, who is headed by Dr. Shashi Alok and Dr. Monika ji, is doing well. So my good wishes for you all. And let's start my presentation first. This screen is visible, my PPT?
Right, sir. PPT is visible, sir. Right, sir.
Okay.
Right, sir. Perfect.
So, very good afternoon to all of you. And myself, Dr. Himanshu Pandey. I am working as director here in CT and as a deputy registrar in the Center Institute of Higher Tibetan Studies, Sarnath, Varanasi. My today's topic is revolution of the nanoparticulate ophthalmic system for targeted and controlled drug delivery. So first of all, I would like to give introduction. You all have might experienced that whenever you are instilling certain drugs into the eyes, because more than 90% of the drug which is used for the treating the eye disorder or disease is available in the topical ocular delivery form. and rest 10% of the drug is either given by the systemic circulation or by the other route. We will discuss it later on. So as far as the topical instillation of the drugs for the treatment of the eye disease or disorder is concerned, you might have experienced that when you will instill one or two drop of the drugs into your eye via topical ocular delivery system, you will experience the number of the drops get drained out. You are instilling one or two drop only, but you will find numbers of the drops get drained out from your eyes. Why it is so? You are instilling only one or two drop, but where comes these numbers of drops which get drained out? The problem is that whenever you instill a drug via through eye drop, your eye considered it as a foreign particles. And since it considered as a foreign particles, our tears production by the reflex action, the eye start producing the tears so as to drain out that particular foreign particles. So due to the lot of tear production, the drug get drained out. And as far as the research report is concerned, only less than 5% of the drug which is instilled through ocular eye drop is get bioavailable for the action. So less than, you can imagine, you are instilling, suppose you are instilling 100 mg of the drug and less than 5 mg is available for the giving the therapeutic action. So due to this reason, the doctor advise for the frequent installation of the drugs into your eyes through ocular eye drop. Not only the tears, but also there are the various reflex mechanism and various drainage system which is available in the eyes, drained your drugs so that the action of the drug is least or the minimal available for the treatment of your eye disease or the disorder. So in the first slide, if we consider, see, you can see the image of the eye. If we consider whole eye, the eye can be divided into two segments. One is the anterior segment and another is the posterior segment. Anterior segment include your aqueous humor, your cornea, your lens, iris. These are the region of the anterior segment, and posterior segment consists of vitreous humor, sclera, retina, optic nerve, blood vessels. All these considered in the posterior part of the eyes, and these are known as the posterior segment. So the disease which is associated with the anterior segment are the conjunctivitis, because in this rainy season conjunctivitis is a very common disease. As far as the Indian scenario is concerned, I am not aware about the other various countries, but as far as the Indian scenario is concerned, because here is the raining season, due to humidity, conjunctivitis is the more common in the village. Apart from that, the disease which is associated with the anterior segment are scleritis, keratitis, uveitis, so and all these thing. And the disease which is associated with the posterior segment are the macular degeneration and age-related macular degeneration, retinitis, and retinopathy. All these are the disease which is associated with the posterior segment of eye. Now, for the treatment of the disease or the disorder of the anterior segment of eye, ocular topical delivery is the very common form of the delivery system. While as far as the posterior segment is concerned, the drug cannot be delivered by the ocular eye drop for the treatment of the disease or the disorder of the posterior segment, because there are various barriers which are available in the anterior segment which restrict the entry of the drug to the posterior segment. So for the treatment of the disease or disorder of the posterior segment, various other routes are available. You can say by the oral delivery, or the much popular route is the intravitreal injection. We will discuss it later on. Yeah, here in next slide I have shown the various traditional route for the delivery of the drug for the treatment of the eye disease. You can see the topical instillation and intracameral, subconjunctival, intravitreal, retrobulbar injection, and systemic. So the problem associate why the ocular topical delivery is the very much common delivery system for the treatment of the eye disorder, because you doesn't require any type of expertise. You have to simply instill the drops into your eye and close the eye, and for the getting the therapeutic effect. It is the another matter that how much drug is getting viable for the therapeutic things. But this is a very common because it doesn't require any expertise. While for the treatment of other certain type of disease, particularly for the posterior segment, we can say the macular degeneration or for retinitis, intravitreal injection is very nowadays it is a very effective. It is not a common, I can say, but I can say it is very effective because we directly instill the drug through injection into the that particular posterior segment. Now, you can imagine how painful this phenomena will be if we are instilling the drug or delivering the drug through injection into the eyeball. So the drawback associated or the limitation associated with the intravitreal injection is that it need expertise. We can say that the each and every doctors or ophthalmologist will able to deliver the drug through intravitreal injection. No, only certain ophthalmologist who has the experience of injecting the drug to the posterior segment can only able to deliver the drug through this intravitreal injection. And nowadays, India is the capital of the, you can say, the type 2 diabetes. And the most common side effect of the diabetes, type 2 diabetes, is the diabetic retinopathy. Okay. So for the treatment of these diabetic retinopathies disorder, generally intravitreal injection, endothelial growth factors, lot of endothelial growth factors are there, which are delivered by these intravitreal injection route of administration. So nowadays it is common in India because the side effect of the diabetes. Now, another route is the systemic administration. Why? By oral route, you can take the drug and the drug will get dissolved and reaches into the systemic circulation, and through the blood, as the eyes get the blood through the systemic circulation, so it is expected that the drug which is available in the systemic circulation will also reaches to the eye for giving the therapeutic effect. But it is not like so, because there is the blood retinal barrier, very strong conjunction of the blood retinal barriers are there into the posterior segment of eye, which restrict the entry of the drug which is there in the systemic circulation to get available for the therapeutic effect. So this route is not very efficient route. Systemic circulation is not very efficient, and intravitreal injection require the expertise, and this is costly affairs. And the topical drug delivery system is the very common. A common man can instill the drug into the eye through the topical administration. So that's why more than 90% of the drug which is available for the treatment of the eye disease are in the form of topical ocular delivery system. Now, here in the next slide, you can see the various route of delivery and their advantages, disadvantages, and the disease which are being treated by this type of things. So, topical, subconjunctival, intravitreal, retroorbital, systemic administration. Okay. You can see the disadvantage of the systemic administration, blood ocular barrier, or you can say most particularly the blood retinal barrier. So low viability, systemic toxicity caused by the, if you able to trying to give the high dose, then they will cause the, it will cause the systemic toxicity. Now, as I had discussed, that when you instill the drugs into the eye, you will find the more than 90% of the drugs get lost by the precorneal activities. One is the tears production, reflex by the reflex action. Once when the any drops get comes into the contact of the ocular surface, the tears production start, and that due to the huge a lot of amount of the tears which is get produced, they wash out the drugs which is instilled by the ocular delivery system due to the simply by the reflex action. That's why you will experience the after instilling 1 or 2 drop, you will get the number of drop which is drained out from your eye. One. Second thing is there is the duct which connects the your nasopharynx and the eye, nasolacrimal duct. That duct is known as the nasolacrimal duct. So when you instill the drugs into the your eye, some of the drug get drained out through the nasolacrimal duct. So nasolacrimal duct you might have experienced when the drug is bitter. After instilling the drugs into your eye through the eye drop, you will experience the taste of the drug into your mouth. How it comes? It is due to the drainage through the nasolacrimal duct. The drugs get drained through the nasolacrimal duct, and through upper palate, it comes into your mouth, and you experience the taste of the drug, bitter taste of the drugs. Secondly, you will be happy to know that the accumulation or the carrying capacity of our open eye in the stage of open, when the eyelids get opened, the drug our eye can accumulate only 30 microliter of the fluid, only 30 microliter of the fluid in at the open stage. But once you blink, that 30 microliter also get drain out, wash out. It get washed out by the blinking. So after installation, when you blink, that rest amount of drug get also drained. So there are and these are the drainage things. But once the suppose the 10% of the drug or the 5% of the drug is available for the therapeutic action, there are the various barriers which is available into the eye, which restrict their entry to the posterior segment. So these are the various parameters or the cause which can restrict the efficacy of the drug which is instilled through the ocular eye drop. Now, recently, to overcome these type of limitations, the scientist has worked out to search some modified, or you can search some novel type of the drug delivery system which can cross out the boundaries of the limitation and able to give the proper therapeutic effect for the effective treatment of the ocular disease or the eye disorders. So one of these is the by using the nanoparticles, we can achieve a lot of the things which can cross the various limitation which I will discuss in the later slide. So by using the nanotechnology, but most of you will be who are young enough and are bachelor student who don't know about the what are the nanotechnology and what are the nano. For them, I had prepared 1-2 slides. Actually, nano is a Greek word which means the dwarf, and it is 10 to the power 9 minus 9 meter. The dimension of the 1 nano is the 10 to the power minus 9 meter. So you can imagine. So in 1,000 nanometer considered as 1 micrometer. So for the biological application of the nanoparticle, it should be between 100 to 200 nanometer of size. But if it is 800, 1000 nanometer, 2000 nanometers, it won't works. It won't works. It comes into the micro range. So to get the application of the nanoparticle, as far as the biological system is concerned, as far as the pharmaceutical system is concerned, we generally restrict the size of the nanoparticles near about or below the 100 nanometers. So, and the science which deals with the nanoparticles are the considered as it comes under the nanoscience. And when we use that particular science to develop certain type of technology, when we are using that nanoparticle to develop for certain type of application or certain type of the technology, that comes under the nanotechnology. That is the difference between nanoscience and nanotechnology. So Why nanotechnology is very prominent nowadays for as far as the pharmaceutical science is concerned, as far as the drug delivery system is concerned. It is due to the mainly due to its size and surface related property. The surface, when we decrease the size of a single big ball into the numerous small nanoparticles, what will happen? The surface volume ratio get increase. The change means the accessible surface area, there will be the enormous increase in the surface area when a big size of the particles get converted into the nanoparticles. So as far as the dissolution is concerned, as far as the surface reaction is concerned, there will be the huge amount of the surface area which is now available by converting the big particle into nanoparticles to take part into the such type of reaction. Optical property of the material get changed, the thermal conductivity, various physical property, melting point, solubility, rheological property, all these get changed by converting the big particle into the nano range. Now here we had developed in our case, next slide you will find that we had developed the nano suspension, controlled release nano suspension for the effective treatment of the ocular disease, particularly of the anterior segment. Okay, so now the question is, what are the control release and how it is get differentiated by the conventional system of the drug delivery system, and what is different between control release and sustained release. So here the drug plasma concentration versus time, you will find the graph. In this graph, in maroon color, red and maroon color, you will find the zigzag shape, and this is the plasma drug concentration versus time profile of the simple conventional therapeutic system, delivery system like tablet or capsules. It reaches the peak, it comes down within the short span of time, within 1 2, it depend upon the T half of the drug, but particularly after 1 or 2 hours it get below the subtherapeutic range. Again the drugs is get administered, means the multiple dosing are preferred in such type of the cases in the conventional tablet and capsule form. In sustained release definitely increase the means the duration of time in the therapeutic level is certainly increased by using the sustained release system, but again it requires the multiple dosing, and the time of the for the therapeutic effect is comparatively low as far as the control release is concerned. But in the case of controlled release, the difference between sustained and controlled release is that both comes under the prolonged release system. Sustained release system simply extend the release of the drug up to the particular period of time. Means you take the any delivery system, suppose oral sustained release system, whether it is the floating tablet or mucoadhesive tablets, it slowly release the drugs. Due to slow release of the drug, the absorption of the drug will be also slow, and the reaching of the drug to the systemic circulation will be the slow and for the longer duration of time. So the drug will be available to certain period of time. But in the case of control release system, it also extend the release of the drug from the delivery system, but with the predetermined rate. Is sustained release, there was no predetermined rate, but in the case of zero order control release, the drug is released with the predetermined rate to achieve the zero order kinetics. And you can see the orange line where you can see the once the drug reaches into the therapeutic level and it maintain the concentration throughout the long duration of time, so that to get the better therapeutic effect. And due to these prolonged release delivery system, the utilization of drug get better, the dosing frequency get low, and the fluctuation of the peaks get minimized. So these are the advantages of the controlled release or the prolonged release system. Here in the advantages, as I said, that total dose is low, reduce undesirable side effect as in the case of the conventional system, reduce dosing frequency, better patient acceptance and why the patient compliance get increased or there is a better patient compliance? Because if you have to take the 3 or 4 tablets a day, you have to remember that, oh, it's now it's 5 o'clock, I have to take the drug. Now it's 10 o'clock, I have to take the drug. Okay. And sometimes the patient get wake up in the early morning at the 5 o'clock or 4 o'clock. No, after 6 hours I have to take the drug. Doctor has said. So now I had taken the drug 10 night. Now it's 4 morning. So I have to take the drug. So there are the various in compliance sort of things with the patient. But when you are using the once a day in the case of the control release system or sustained release system, once a day drug, you simply in the morning you take a drug and you get there is no headache, no relief. You will get relief throughout the day. There is no confusion of taking the drugs and remembering that I have to take the drug or not. Okay. And less fluctuation at the plasma drug level, as I have shown in the figure, that in the case of the conventional system, you will find the more fluctuations. The uniformity of the drugs is there, and definitely due to these things, the efficacy and safety ratio get increased. Now, it is not always there that control release or the prolonged release systems are better systems always in all the case. No, they can be preferred in the certain case. There are certain parameters which has to be taken into consideration while developing such type of the prolonged release system. Because their disadvantages or the limitation associated with such systems are very much important. Is the dose dumping. What is the dose dumping? Dose dumping is the you had encountered or incorporated the drugs in to in such a way that the drug will release slowly and slowly. What will happen due to the certain reason, the drug get abruptly diffused out from that particular your delivery system, your carrier. Due to abrupt release of the drug, the concentration of the drug into your blood will shoot out into the toxic concentration. So this phenomena is the dose dumping, means the abrupt release of the whole of the drug which has been loaded into the carrier into the biological system. जैसे biological system में वो आएगा, it will cause the dose dumping. So this is the one of the disadvantages or limitation. फिर next is the for if you are giving the certain control release system or the any sustained release system drug to the patient, additional patient counseling is required, because in sometimes the carrier in which the drug is loaded is coated with the polymer, and that particular polymer is responsible for releasing or controlling the release of the drug. But if the polymer get detached or the because polymer are intact, if certain due to the certain reason, if there is any the integrity of the polymer get lost, then what will happen? The drug get started abruptly. There will be the no control. In certain case, in Indian scenario particularly, if the doctors are giving a drug so that you have to take once a day, so the patient may due to suppose the patient is elderly patient, so there he have the problem for engulfing the drug with the water, the huge amount of the tablet of the capsule or any other delivery system. He or she will try to break it into the small piece, and he or she will feel that now the particle of the drug is get small and I can easily swallow that particular system. So it is not advisable at all in the case where the coating is there to control the release of the drug. Once coating get deteriorated or disturbed or defragmented, there is no way to release the control of the drug. All the drug will be available for the biological system. So they may lead to the toxic concentration. So additional patient counseling is required for those patient who has been given the prolonged release system or the drugs. Now, in certain liquid sustained release formulation, stability problems are there due to the various formulation parameters and considering the taking the polymer to control the release of the drug. Sometimes they are not very cost effective. They are costlier than the conventional system. And as far as the antibiotics are the concerned, they develop the bacteria get develop the tolerance against that particular drugs if they are given in the sustained release or prolonged release formulation. So you will find that as far as the antibiotics are concerned, you will very rarely find the controlled release system of the antibiotics. Antibiotics are generally given in the immediate system, not control or prolonged release system. You won't find, because due to this reason, the microbes get develop the tolerance against that particular drug. That is the reason. Next. So in my case, we had in our lab developed the gentamicin loaded nanoparticles. Gentamicin is a well available drug which is used generally for the treatment of the top anterior, the disease of the eye associated with the anterior segment, generally the ocular infections of the anterior segment. So gentamicin is a drug aminoglycosides, and they are particularly they are used against the gram negative bacteria, and particularly it has been selected here, so because the various gram negative strains, resistant strains, the gentamicin is very effective. So we had taken the gentamicin as our model drug, and we had used the polymer Udrazid RL RS100, and they are the copolymers of the ethyl methacrylate and acrylate. They are because it has been selected because they are able to swell. These are insoluble in physiological pH, and they are able to swell. So we had prepared the multiple emulsification. We had prepared the drug loaded nanoparticle by the multiple emulsification techniques. First of all, you by the flow chart you can see we had used the 5% weight by volume solution of gentamicin sulfate, and it has been slowly by the syringe, drop by drop, it is dissolved in the dispersed in the methylene chloride 20 ml containing the polymer Udrazid RL RS100, and we had sonicated it at 60 seconds at 40 watt, and after that we will get the water in oil emulsion. This water in oil emulsion is again by simply drop by drop by the syringe injecting into the 25 ml of 1% polyvinyl alcohol solution, and again it get emulsified for by the sonication, and then water in oil in water emulsion will form, and in the to make it into the nano emulsion, nano range homogenization has been done at 60 bar for the C cycle, and it has been again diluted with the 100 ml of PVA solution. And after evaporating the internal phase, the rigidization process takes place, and the polymer drug nanoparticles get obtained, and we had lyophilized it by using the mannitol as a cryoprotectant. And you can see in the figure 1A and B represent the gentamicin solution in water and the gentamicin nano suspension, and the lyophilized gentamicin loaded nanoparticle in the figure 2. We had characterized the various prepared nanoparticle by the scanning electron microscopy, transition electron microscopy, and atomic force microscopy. The SEM, TEM, and AFM images are here, and their crystalline structures has been analyzed by the thermogrammetric analysis, differential scanning calorimetry, and X-ray depression. Zeta potential, polydispersity index, and surface charge has also been evaluated. Here you can see the thermogrammetry analysis chart. The green showing the thermogrammetry weight loss versus temperature of the gentamicin sulfate. Red line, the pure, red line is the Udrazid and gentamicin sulfate both composite, and blue is the pure Udrazid. So you will find the stability of the composite of the Udrazid and gentamicin sulfate is near about, they can having their integrity up to 225 degrees centigrade. After that they get decomposed, but as far as the single gentamicin is concerned, the degradation start very at near more than 75 degrees temperature, and as far as the pure Udrazid is concerned, it is also start gradually degrading with the increase in temperature. Here, the differential scanning colorimetry analysis were used to characterize the thermal behavior of the polymer of the Udrazid RS RL100 gentamicin sulfate. So you can find the various composition of the means the dropout of the heat flow at phase 1, phase 2, and which is given for the different gentamicin sulfate, and the red is for the Udrazid GAVO. So we can say that they are the in the case of the polymer nanoparticles with the Udrazid, there is the higher degree of the crystallinity of the polymer blend, thereby considered at the for the better storage stability. Now, the drug which has been incorporated, the drug means the gentamicin, which has been loaded into the polymer, the encapsulation efficiency has been determined by this formula: total gentamicin minus free gentamicin after total into 100. And we had prepared the 5 formulation having the different drug polymer ratio. And with these, the particle size you can find for the GN1, the particle size is 145 plus minus 18.3. The average particle size is 145. Zeta potential is 26. Near about polydispersity. What is the polydispersity index? Polydispersity index is the parameter which is determined to know whether the nanoparticle which has been prepared are uniform in size or not. Suppose the average particle size you will get, suppose it is 150 nanometer, okay, but there is the chances that more than 20% of the particle is more than 500 nanometer of size. Few particles are 80 nanometers. So there is the various variation in the particle size. Although the average particle size is 150, but there is the chances of the variation in the particle size. This polydispersity index signifies the uniformity in the particle size. Okay, the average particle size is 150 and the and it is uniform. The lessest particle of 120 nanoparticle and the highest one is of 180 or 190. So it is acceptable. But if the polydispersity index is more, so it's mean there is the non-uniformity in the particle size and it cannot be suitable for the biological purposes. Here, with the GN1 formulation, you will find that the encapsulation efficiency of 81%, which is considered as good, and release efficiency for 12 hours, we had performed the release profile, and it has been released about 92% of the drug is get released by this formulation. Here you can see the graph of the release profile of the drugs, and for GN1 formulation, after 12 hours, more 92% of the drug has been released, while with the other formulation, the total release is not get achieved. Now, okay, it gets released, but what are the mechanism of the release of the drugs from the polymer? So for this, we had put up the values into the various models: zero order, first order, Korsmeyer, PPAS, and Higuchi. And we had find out that the value of the N, which is near about approaches to less than 0.45. It is, if it is less than 0.45, it is considered as the drug is following the simple diffusion. You can say the Fickian diffusion process for the release. Here we had performed the stability studies at different temperature for the 6 month for the various formulation, original nanosulfuration and freeze-dried nanoparticles, and it has been found that after 6 months the stability is very acceptable. You can see the data of the drug loading and the minimum size. With the GN1 formulation, after 6 months at 25 degree and 60 degree relative humidity, the mean size was 189. Initially, it was 145. And for the freeze-dried nanoparticles, initially it was average was 152, and after 6 months it is 205. And the loading capacity, which was initial value of 81, after 6 months, the very insignificant degradation of the drugs has taken place. And you can see the result. Now, the nanosuspension of the gentamicin loaded nanoparticle has been developed. Whether it is acceptable by the biological system or not. For that, we had performed the animal experiments in the rabbit, and we had instilled the drugs into the rabbit eye, and we had count to the winking of the rabbit eyelid. Normally in 1 minute, the rabbit generally wink for one times. So 1 to 2 per minute winking. So for the group 1, where is the control, only simulated tear fluids of PS 7.4 was given. The winking count was 8. And in the case of the commercial eye drop gentamicin sulfate, it was 7.33. And in the case of nano suspension, it was 10. So if we consider the normal winking rate is 1 to 2 per minute, so for 5 minutes, 10 winking is acceptable. So we can say that the our drug nano suspension has passed the ocular irritation test. There is no significant irritation is there in the biological system. Now, as far as the viability is the concern, we had also performed the viability study, the Cmax of the gentamicin nanosuspension and the gentamicin sulfate solution, which is there. You can see the viability Cmax of our drugs, which is instilled into the eye of the rabbit, is 14.95. Very significant increase, while in the gentamicin sulfate solution, where I have said it get drain out, and very less amount of drug is available for the biological action, you can find out here the area under the curve for the means the viability of our nano suspension is 87.34, while the viability for the gentamicin sulfate solution is 38.14. How significant increase in the viability is. Here you can see the treating the infected eye of the rabbit. Here you can see the image A of the controlled normal eye of A, F, and K are the photographs of the normal eye of the rabbit. A is of the control, F is of the gentamicin solution, and K is for the nano suspension. Now, after day 0, you will find this is the normal eye, and day 0 is the infected eye. You can find the various mucus is getting drained out because eye get infected, it get red. And day 2, from day 0, we had started the delivering the drug, giving the therapy by instilling the drugs. You will find day 2 there is no significant improve, day 4 slight improve, and day 6, after day 6 with the nano suspension, you can see there is no redness in the eye and there is no secretion in the eye, you can see. But with the gentamicin solution at the day 6, yes, there is the relief in the eye, but as compared to the nano suspension, it is not, still there is means the secretion is there available, and the slight redness is also there. So we can say by this animal experimentation that if the drug is incorporated in the nano suspension, and we had seen the viability of the drug get increased, so there is the significant increase in the therapeutic effect also. So the significant outcomes is the improve the pharmacokinetic and biodistribution of the therapeutic agent for the intended side, reduce ocular toxicity. Certainly, the dosing frequency get decreased due to control release system, and lower dose is required for efficacy. Increase in stability, wide variety of the therapeutic agent as the drug is proposed to encapsulate into the nanosystem, and focus on the possible economical production on large industrial scale, and to increase the stability of the developed system during storage. So these are about my presentation nowadays. So if there is any question from the audience, so you can definitely, you all are welcome.
Thank you so much, Dr. Himanshu Pandey. Thank you so much, sir, for delivering such an enlightening and insightful session on revolutionizing eye care, nanoparticulate system for enhanced ocular drug delivery. Your deep knowledge and clear presentation made complex concepts accessible and engaging for all the participants. We sincerely appreciate your valuable time and contribution to the 96th SPSR webinar, and we are grateful for the opportunity to learn from your expertise. Thank you so much, sir. Now, sir, we will ask some questions from the participant side, and I will be asking you the questions one by one. The first one is, sir, why is gentamicin sulfate a suitable candidate for nanoparticulate delivery in ocular treatment?
Am I audible?
Right, sir. Audible, sir.
Okay. So there was any hindrance during my presentation, or it was streamlined? Okay.
It was streamlined, sir. Flawless.
Okay.
Wonderful, sir.
Okay. 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 Any question from the audience?
Sir, the first one is, why is gentamicin sulfate a suitable candidate for nanoparticulate delivery in ocular treatment?
Yeah, actually, I had considered taking this gentamicin because due to three reasons. First of all, it is readily available, and these drugs is very commonly used for the treatment of the ocular infection, particularly associated with the anterior segment. So it was very means simply we can compare our nanoparticle formulation of gentamicin with the available marketed solution. One thing. Second,
Right,
the
sir.
it is used for the treatment of the gram-negative bacteria, and gram-negative bacteria are widely cause the ocular infection. So this was the second reason why we had chooses this drug. Secondly, the elimination half-life of the gentamicin is 2 to 3 hours. So the elimination
Right,
half-life
sir.
of 2 to 4 hours is very suitable for the preparation of the control release formulation. So as we had made the control release formulation, we have selected this gentamicin as a model drug.
Right, sir. Thank you so much, sir. The second question is, sir, how does the drug to polymer ratio influence the encapsulation efficiency and release profile?
Definitely, as I had shown in the slides, when we are increasing the concentration of the polymers, so the release of the drugs, as the polymer is swellable polymer, they are the copolymers, and they are cross-linked with each other. So when the polymer concentration get increased in respect of the drugs, they restrict the release of the drugs from that particular their arrangement, means the chemical arrangement. And
Right.
while having the polymer concentration less, the drug can be easily released from that particular drugs, and it can be achieved.
Right, sir. Thank you so much, sir. The next one is, sir, how does the pharmacokinetic profile of your nanoparticle system compare with the commercial gentamicin eye drops?
The same I had shown in our slides also. Actually, the gentamicin solution which is available commercially in the market, that's all, not only the gentamicin, but any solution of the ocular eye drops having the less bioavailability due to the drainage system and the restriction by the biological barrier which is present in the eye. So the viability of such type of drug which is available in the simply traditional solution form will having the less viability, and if the viability will be the less, definitely the therapeutic effect will be the less. While we had used it the carrier mediated system, where we had using the carrier of the polymer which is mucoadhesive, that polymer having the positive charge, because we had chosen this hydrazine polymer due to having their positive surface charge, because the mucin layer of the eye, the corneal mucin are anionic in nature, they are having the negative charge. So there will be the proper interaction and binding with the positive and negative charge, so that particular polymer has been used. And by using that particular polymer, the residence time of the drug carriers was increased several times as far as the conventional marketed commercial available solution. So due to increase in the residence time, the availability of the drug are more and therapeutic effects are more.
Right, sir. Thank you so much, sir. Next one is, sir, how stable are the nanoparticles under different storage conditions in your stability studies?
As compared with the marketed preparation, we know that for the degradation of the drug, oxidation, hydrolysis, and photolysis. These are the three main criteria which degrade the drugs. So when the drug is exposed into the aqueous solution, as the commercial eye drop is there of the gentamicin, the hydrolysis of the drugs is very frequent and the drug get decomposed readily as compared. But here in our case, we had incorporated our drug into the carrier system. We had not exposed the drug available to the aqueous media. So the drug were well encapsulated into the polymeric particles, and therefore the stability of our drug will be much more, significantly much more than that of the commercial ocular solution.
Thank you so much, sir. The last one is, sir, are there any plans for the scale-up and clinical trials for this nano formulation?
We had already during the our once I complete this our lab mates complete this reactions and made the formulation successfully, and completed all the animal experimentation and pre-formulation study. We had approached to the micro labs. Our guide had approached to the micro labs. There was some initial talks has taken place, but it has been not reaches to such conclusion to have the market exploration of that particular formulation. But definitely we can do that. Definitely we can do that.
Right, sir. Thank you so much, sir, for answering all the questions so wonderfully. Your insights into the nanoparticular ocular drug delivery system were not only enlightening, but also thought-provoking. We truly appreciate your time, expertise, and the clarity with which you shared the complex and cutting-edge topic. It was indeed a privilege to learn from your experience, and we are confident that today's session has added a great value to all our attendees. We look forward to more such impactful engagements with you in future. Thank you once again, sir. So here we
Thank you
come
so
to the
much for
conclusion.
providing me this opportunity. Thank you, Dr. Shashi Alok ji, Monika ji, and very thank you, Dr. Pradeep ji, for having such a nice proceeding of this talk. And I extend my best wishes, good wishes for your society, and may God bless you all. Thank you.
Thank you so much, sir. It's our privilege, sir. So here we come to the conclusion of this session, sir. So 96th SPSR webinar on behalf of the Society of Pharmaceutical Sciences and Research, I extend our heartfelt thanks to Dr. Himanshu Pandey for delivering such a insightful and engaging session. Your expertise on nanoparticulate system and their role in enhancing ocular drug delivery system has provided our audience with a deeper understanding of emerging trends in the pharmaceutical innovation. We are also sincerely thankful to all the attendees from across the globe for your active participation and thoughtful questions. Your enthusiasm continues to inspire us to organize more such enriching academic sessions. On behalf of the Society of Pharmaceutical Sciences and Research, we sincerely thank Dr. Himanshu Pandey for his valuable contribution and all participants for their active engagement. We are deeply thankful to Dr. Shashi Alok, the National President of SPSR, for honoring us with his presence. We recognize and appreciate your commitment and guidance, and we offer our profound thanks for your significant input. We would also like to express our appreciation to our convener, Mrs. Monika Sabharwal, for her pivotal role in organizing this event. We extend our gratitude to all the SPSR team members for their collaborative efforts, which have broadened our understanding and fostered cooperative learning. Your collective contributions have made this webinar a success, and we look forward to continued collaboration in advancing pharmaceutical sciences and research. We appreciate your interest in SPSR webinars and hope you will join us for future sessions exploring innovative topics in pharmaceutical sciences. Thank you, and we look forward to seeing you at our upcoming events. Stay connected with SPSR for upcoming webinars and initiatives. Until then, take care, stay safe, and continue the journey of lifelong learning. Thank you and have a wonderful day ahead. Thank you so much.